My name is Cale Gushulak. I'm a PhD candidate in the Paleoecological Environmental Assessment and Research Laboratory in the Biology Department at Queen's University. My project examines the climate of northeastern Ontario over the last 8,000 years.
Approximately 9 to 5,000 years ago, the temperatures across North America were 2 to 6 degrees higher than present. This time is known as the Holocene Thermal Maximum, or HTM, but it did not occur evenly across the continent as some regions became warmer earlier than others. This project attempts to discover the specific timing and magnitude of the HTM in northeastern Ontario using paleolimnology, which is an interdisciplinary science that examines the physical, chemical, and biological data preserved in lake sediments. Sediment is collected in sediment cores. Here is a demonstration of a core being taken. Note that in my project this is done on a much larger scale. The core is dropped into the lake on a line, with the core tube sinking into the sediment. A weight is sent down the line, triggering the mechanism which seals the tube, and then the whole device is raised on the line back to the surface. Here the core can be collected and the sediment can be analyzed for various proxies.
Do you want to talk about what you're doing right now?
Sure. Right now I'm using a Weson meter to measure the temperature and the amount of oxygen in the water going down in segments to its depth. So we're going to use the length of this cord, which is 15 meters, and then we're going to use the Van Doren apparatus to do some depths below that. With that information, we'll be able to tell the way that the lake stratifies during the highest point of the summer.
Back in the lab, sediments are stored in a 4-degree cold room in discrete samples within these bins. Samples can be subsectioned for biological proxies or to be sent for radioisotopic dating. Support from the W. Garfield Weston Fellowship through WCS Canada allowed us to send more samples for high-resolution radiocarbon dating, which will allow us to have a more precise understanding of the timing of the Holocene Thermal Maximum in Ontario's northern boreal forest. To isolate biological microfossils, sediments are digested in strong chemicals. Safety is always important when using such powerful acids. Once processing is complete, we are left with slurries composed of the material that we want to examine. These vials are filled of siliceous materials, which will be composed of diatom and chrysophyte algae. The slurries are then placed on microscope cover slips. These will be allowed to dry and then permanently mounted to make microscope slides. Microfossils are counted and identified on the microscope. Data collected in this way is then analyzed using various statistical methods. To obtain reliability in results, hundreds of fossils must be counted per slide, so my lab mates and I spend many hours on the scope. There are two main biological proxies which are examined in my project. The first are diatoms, which are a group of siliceous algae. Specific taxa have known optima and tolerances to various chemical and physical characteristics, so by examining how the diatom flora changes through time, we can interpret past environmental changes such as nutrient levels, water levels, or pH. I also examine terrestrial pollen to track changes in forest ecology and overall climate. This provides a climatic framework for more specific limnological changes. By reconstructing the environmental and climatic conditions of the Northeast Ontario region during the HTM, we hope to be able to better understand and predict how this region may change in response to future climate warming. I would like to again thank the W. Garfield Western Foundation and WCS Canada for their support. They have allowed us to expand our project beyond its original goals.